Hello, anatomy friends. This is Dr. Alsip, and we are going to start exploring the wonderful world of skeletal muscle tissue at a more microscopic level. So we're going to focus on skeletal muscle fibers, which goes by many names. The scientific name is going to be myofibers, but we'll talk about some others as we go through. So we have much to do. This is going to be one of the longer videos as we're going to discuss, first of all, how they even form, but then we're going to spend a little bit more time in terms of the structures within a myofiber. And last but not least, we're just going to introduce sliding filament mechanism. Very physiology heavy, so we're not going to get into the details here, but it's super important in terms of understanding how muscles actually contract. All right, excellent. So during embryonic development, hundreds of myoblasts, which are just these small mesodermal cells. Remember, mesoderm is one of the germ layers so you'll have these mesodermal cells that will collect. As you can kind of see here, they start off individually, they start to collect and fuse to form these long muscle fibers, like you can see here. And that is why each mature muscle cell or myofiber will have hundreds of nuclei associated with them because they are from these different cells that have kind of formed together. So we have multinucleate cells. But once a muscle cell is fully fused and considered a mature cell, cell division is not possible after that point. But we do know that muscles grow, right? We know that muscles get larger. So how do they do that? Well, in order for a muscle cell to get larger, it has to what's referred to as hypertrophy, which is the enlargement of the existing muscle fibers. So this hypertrophy is possible by the increased production of structures in the cell. Most noticeably, what often gets larger or what often increases are the amount of myofibrils, and we'll talk about what that is in a moment. But you can have other components of the cells that will increase, you'll have more of. So that could be the sarcoplasm, the sarcoplasmic reticulum, mitochondria, other organelles. Those can also have increased production, therefore making the cell get larger. Now, hypertrophy, not hyperplasia. Okay, so hyperplasia would be more cells. Hypertrophy is the increase in size of the existing cells. During childhood, certain hormones like human growth hormone, like we talked about before, or sex hormones can lead to hypertrophy of those existing cells. But after the juvenile period, muscle hypertrophy really generally can occur due to repeated forceful muscular activity like weight training that can lead to a larger size of existing muscle cells. So that's a little bit about how muscles will actually grow. Now let's return back to this concept of the myofiber in order to better understand the structures within. So we mentioned some of those names, so let's better understand what they are. Now, as I mentioned at the beginning, muscle cells go by other names. I personally like the name muscle fiber. You'll hear me say that quite a bit because I think it's indicative of this long size of one muscle cell. It's a really long cell compared to other types of cells. Again, myofiber is the more scientific name, and sometimes you just hear it referred to simply as the muscle cell. And as you can see in this image, there's a lot going on in one cell. It's like packed full of stuff. So what is that stuff? So let's talk about that. Most of the structures within a muscle cell have slight variations to their names to indicate that they are part of a muscle cell. So there are a lot of the same components that you have in all cells, but oftentimes you'll see it with this sarc associated with it, which means flesh. So if you see this sarc, generally you can think muscle cells or part of a muscle cell. So let's start with the sarcolemma. And the sarcolemma has two parts. Sarc means flesh, lemma means sheath, and basically, in essence, the sarcolemma is the plasma membrane of a muscle cell. All right, so you can see that kind of in this cartoonish image here surrounding the contents of the muscle cell. Now, in certain areas of the sarcolemma will invaginate or tunnel into the surface and go into the inner contents of the cell. And this is referred to as a transverse or T-tubule. And these things are super cool, very important in terms of contraction, because these invaginations allow muscle action potentials to propagate or spread widely and, importantly, quickly throughout the muscle fiber. So not only will that action potential kind of be traveling along here, it will actually go down into the inner portion of the cell. And one thing, let me go back to the slide, and you'll see in this other image, is really closely related to this other structure within the cell called the sarcoplasmic reticulum. So a little bit about what the sarcoplasmic reticulum is, or the SR. These are fluid-filled membranous sacs that encircle each of the myofibrils. Now, myofibrils are the major structure within the myofiber. And that's going to be those contractile structures. More to come on that in a moment. The sarcoplasmic reticulum is similar to the smooth endoplasmic reticulum in non-muscle cells. And key to the function of the SR is that it stores, and where do I have it here? Right here. It stores calcium in relaxed muscle. That's a huge part of its importance functionally. Now note, I said it's in a relaxed muscle. It stores in a relaxed, and in a muscle cell that has been activated, this will be released, and that's very important. At the ends of the sarcoplasmic reticulum, there are these dilations of the reticulum called terminal cisternae. You can see one here, you can see one here. And what is it really closely related to? It's related to that transverse T-tubule. It's right next to it. In fact, one T-tubule and two terminal cisterns equal a triad. So you'll hear a triad associated with the muscle cell. And what can happen is that action potential will travel down that T-tubule, and it will trigger the release of that calcium that we know is stored in the sarcoplasmic reticulum in a relaxed muscle. But that action potential is allowing for the muscle to be activated. And that will allow the calcium to release into the sarcoplasm of the cell to allow muscle contraction to occur. Okay, so we are going to get into the other components. So we talked about the sarcoplasmic reticulum, we talked about the sarcolemma, and then we're going to have this, we're going to have portions of the other contents as well. So you have the sarcoplasm, which is basically the cytoplasm of the muscle cell. And there's a lot going on in there. There's that sarcoplasmic reticulum, as I mentioned. You're also going to have other components in there as well. The bulk of the contents of the sarcoplasm are these structures called myofibrils, and that's what these are all over the place right here. So it's these threads basically that are filling up. This one has been pulled out and then magnified right here. We're going to come back to the myofibrils in the next slide because this is really pretty important. But let's talk about these other structures. There's obviously going to be mitochondria associated with the sarcoplasm, very important in terms of ATP production. There is also myoglobin, not really shown in this image. Myoglobin, these are red-colored proteins that are only found in muscle. And these proteins bind oxygen molecules that can diffuse into muscle fibers from the interstitial fluid. And this oxygen will be released from myoglobin when it's needed by the mitochondria for ATP production. So very important in terms of ATP production. The sarcoplasm also has a substantial amount of glycogen, again, not really shown in these images here. And glycogen is a molecule that stores linked glucose molecules that also can be broken down when needed to help produce ATP. ATP keeps coming up over and over again. So ATP, if you haven't heard of it before. And this is going to play, this is an organic compound that plays a role in energy transfer, and it's vital to allowing muscle contraction to occur. So it's allowing muscle contractions to actually happen, but it also allows for muscle contraction to stop. You need ATP for both. So hugely, hugely important. So say, particularly if you have like sustained contractions over time, you need a lot of ATP for that to happen. Okay, returning to those myofibrils, so all these little things that are dotting throughout the entirety of that sarcoplasm. These are the contractile elements of the muscle cell, and one myofibril will extend the entire length of a muscle fiber. Okay, so they're long like the muscle fiber as well. And they have these prominent striations, as you can see in this kind of close-up illustration right here, that gives that very prominent striped appearance of the muscle cell. So when we talk about striated muscle, myofibrils are playing a role here. Now, these myofibrils can be broken into even smaller structures called myofilaments. And these myofilaments are proteins, and there's three main types associated with a muscle cell. You're going to have contractile, which we'll spend a little bit more time on, regulatory, which will kind of turn this on and off, and structural. And we're not going to spend as much time on the structural components of the myofilaments, but they are very important in terms of allowing for the elasticity and the extensibility that's very vital as well. But as we don't have a whole course to talk about these things, we're going to focus in a little bit more on these other structures. All right, there are two main types of contractile protein. There is actin and there is myosin. And what these proteins do is convert ATP's chemical energy. So it's converting chemical energy into the actual mechanical energy of motion. Myosin, which you can see a myosin molecule right here, is going to be the main component of thick filament. I should say thick filament here instead of protein. Actin is going to be the main component of thin filament, or the thin filament, or a bunch of proteins making up the filament. So all these little things, all these little balls right here basically are supposed to be representing actin. And this, you're going to have two thin filaments for every thick filament. And these thin filaments will overlap with the thick filament during various phases of muscle contraction. Also, as you can see a little bit better on this, this image here, each actin protein is going to have a myosin binding site. So that's what these are representing right here. And that will allow the myosin head to actually attach to the actin during the contraction process. Another component of the thin filament, so obviously actin is going to be the major component, but you're also going to have regulatory proteins that are playing a role as well. So in a relaxed muscle, or a muscle that's not contracting, myosin is blocked from the myosin binding sites on actin, as you can see here, by tropomyosin. Okay, so tropomyosin is going to be this fairly large protein. You can see it kind of sitting right here and blocking the myosin from being able to bind to the actin. Now, also very closely associated to the tropomyosin are these troponin molecules. It's going to keep that tropomyosin in place, but when you have a calcium ion that kind of moves in from that sarcoplasmic reticulum and binds to the troponin, it will cause there to be a change in shape and the movement of that tropomyosin away from those myosin binding sites to allow for the process of contraction to occur. When there's not calcium in this region and that kind of moves out of the way, that tropomyosin will move back into place and block these sites. So that's why these are called regulatory proteins. So the organization of these structures are into these basic functional units that are called sarcomeres. On each end of the sarcomere, you will have these Z discs. So you can see one here, you can see one here, and basically that space in between, that is one sarcomere. And how much overlap there is between the thin and the thick filament depends on whether the muscle is relaxed, stretched, or contracted. Don't worry about these H zones, A bands, I bands. They're super cool. If you ever take a physiology class, you're going to spend a lot more time in these regions here, but we're not doing that deep dive. But you can see in this illustration, you see that representation of the thin filament and the thick filament. So there's 2 thin filaments per thick filament. For our purposes in this course, I will be happy if you understand that skeletal muscle can shorten during contraction, but that the thick and the thin filaments don't necessarily shorten. They just slide past one another. So the myofilaments don't shorten or contract, they slide. But you can see in this image that the thin filament is going to slide over that thick filament, and it causes the sarcomere to shorten. So here's a relaxed muscle. Here's a contracting muscle. You can see here's a thin filament. Here's a thick filament right here. And there's not that level of overlap right here, but during the contraction, that thin filament will slide over. And what that does is it brings the Z disc with it, causing the sarcomere to shorten. And generally, sarcomeres will shorten at the same time to cause for overall shortening of the muscle, of the whole muscle. Okay, so obviously that is a very simplistic version of the physiology of a skeletal muscle. But some of these concepts will come up again, particularly in some of these proteins. But basics I want you to understand are that obviously we see that skeletal muscle can shorten with concentric contraction, but it's not necessarily these structures that are. They're kind of sliding over one another, but the connective tissues and such can play a role as well. All right, excellent. My favorite time is question time, right? All right, so the question is, which structure of a myofiber stores calcium in a relaxed muscle? All right, so pause the video if you need time, and when you're ready, let's look at this question stem again. So a myofiber, that's that muscle cell. What stores, so stores is a keyword, calcium, and another key here is in a relaxed muscle. All right, well, we didn't talk about dystrophin. That's actually one of those structural proteins that we didn't get into. So it's not dystrophin. Generally, if you don't know, if you've never heard of a term, then generally it's not one you need to focus on in terms of the question. The sarcoplasm certainly can have calcium within it, but generally it is going to be in the sarcoplasm and affecting the myofilaments and attaching to particular proteins in a muscle that is beginning to contract. Sarcolemma is that plasma membrane around. There's calcium going on there, but it's not so much stored there. And then actin is going to be a major component of the thin filament, but not so much that. It is going to be the sarcoplasmic reticulum that is going to be hugely important in terms of storage of calcium in a relaxed muscle. If it gets the message or the action potential allows for the release of calcium into the sarcoplasm, that's generally when you begin the process of muscle contraction. All right, excellent. Thank you so much for this big picture view of the myofiber and some of its smaller contents within. This is really interesting area. Now we're going to move into what are the different types of muscle contraction. Oftentimes we think of concentric contraction and shortening, but there's others as well that are as important. This has been a lot of fun, and I look forward to seeing you in the next video. Thanks.